So continuing with the xylem structure, the xylem is a complex tissue, so it means that it contains more than one type of cell. It has the parenchymal cells, the fibers, and water conducting cells. There are two types of water conducting cells. The xylem of all vascular plants contain the tracheids with pits, which allows the water to flow. And in angiosperms, the xylem also contains vessel elements with perforations for the transport of water. Perforations are the openings in the end walls that lack both primary and secondary cell walls. And so when you consider hardwood versus softwood, that's not really telling you how hard the wood is, because both of the woods are going to be hard. But what it's indicating is whether or not the wood contains vessel elements or if it only contains the tracheids. So if it does contain the vessel elements, it's considered hardwood. And if it only contains the tracheids, then it's considered softwood. Okay, so the tracheids would be things like some of the gymnosperms. The ones that contain vessel elements and tracheids are the angiosperms. Okay. So here you can see the tracheids, which are long, tapered cells with pits, which allows the water to flow not only up, but also across other tracheids. And the vessel elements are shorter and wider and have the perforations as well as the tips. And it also has the same function. It allows for the water for water to for what it's involved in water transport. The phloem is also a complex tissue. It contains the two types of cells. So there's the sieve tube elements, which are long, thin cells with perforated ends called the sieve plates. And then there's the companion cells that maintain and support the sieve tube elements. Okay, because the sieve tube elements lack nuclei and are directly connected to the companion cells via the plasmodesmata, okay, which was that structure that connects the two cells. Or it's plasmodesmata if you're talking about more than one, but if you're only talking about one specific structure, it's plasmodesma. Okay, so here are the sieve tube elements. Okay, we have the sieve plate, and then we have the companion cells adjacent to the sieve tube elements. So primary growth allows plants to extend in length. Okay? So because plants grow throughout their lives, they have to have these meristems. And meristems are populations of undifferentiated cells that retain the ability to undergo mitosis. Apical meristems are meristems that are found at the tip of either the root or the shoot. And those are the ones responsible for primary growth. So again, primary growth meaning lengthening the plant. The cells and tissues that are derived from the apical meristems make up the primary plant body. So here you can see the apical meristem. So here is where the new cells are created. And then we see where the different tissue types develop. So we have the procambium. From the procambium, you get the vascular tissue. From the ground meristem, you get the ground tissue, and from the protoderm, you get the dermal tissue. Okay. So here are the the things that the apical meristems gives rise to. So there's the protoderm, ground meristem, and the procambium. Okay. Protoderm gives rise to the dermal tissue, ground meristem gives rise to ground tissue, procambium gives rise to vascular tissue. And here, when you look at a cross section, if you're looking at at the sh- um, the stem of the plant, you can see the ground tissue is in the middle, okay, as well as outside of the vascular tissue. So in lab, we did go over the vascular bundles. So when you have the xylem and the phloem adjacent to each other, okay, those are the vascular bundles that are made from which are types of vascular tissues. Then we also have things like the pith and the cortex. That's the ground tissue. And the epidermis is part of the dermal tissue. The root apical meristem is protected by a group of cells that are called the root cap. And these are cells that are produced by the meristem, and they constantly replenish the cap, okay, because it loses cells. The root cap is able to sense gravity to determine the direction that it's going to grow, and it also secretes lubricant to reduce the friction as it's pushing through the soil. So there's three distinct populations of cells that are found behind the root cap. There's the zone of cellular division, which contains the apical meristem as well as the protoderm, the ground meristem, and the procambium. And then there's the cells in the zone of cellular elongation, which is the one that allows it to increase in length. There's the cells in the zone of cellular maturation, and these are the ones that differentiate into the dermal, vascular, and ground tissue. And they're able to absorb water and nutrients through the root hairs. Okay, so these are the root hairs. And when you actually, well, there's a diagram, this is the actual picture. So these are the root hairs. So how is the primary shoot organized? The arrangement of the vascular bundles and ground tissue in the stem differs between monocots and eudicots. In a eudicot, the vascular bundles are arranged in a ring near the stem's perimeter, and the ground tissue is divided into the central pith in the middle, with the cortex surrounding the vascular bundles. In a monocot, the vascular bundles are scattered throughout the ground tissue. Okay, so here is that ring of the eudicot. So remember that eudicot, same thing as dicot. Okay, eudicot just means true dicot, because remember a dicot is a paraphyletic group. Okay. So here are those vascular bundles. There's the pith. The cortex is outside of the vascular bundles. Okay. And then for the cross section of the monocot stem, these are just scattered throughout. The vascular bundles are scattered throughout. And you also have ground tissue here, but because there's no specific regions, uh, it's just called ground tissue here. Okay. So we said that primary growth increases the length, Secondary growth increases the width of roots and shoots. So this also increases the amount of conducting tissue that's available, and it provides additional support. The secondary growth is the one that produces the wood, and it occurs in the species that have a cambium in addition to the apical meristems. So in lab, we did go over the vascular cambium that's positioned between the xylem and the phloem. So this differs from the apical meristem because it forms a cylinder that runs the length of the root, trunk, or branch. And when the cells divide, it increases the width of the plant. So there are two types of cambia in plants. There's the vascular cambia located between the secondary xylem and the secondary phloem. And then there's the cork cambium, and this is located near the outer perimeter of the root, trunk, or branch. Okay. So in the stem of a woody eudicot, the single layer of cells between the xylem and the phloem becomes the meristematic and forms the vascular cambium. Another ring of cells under the epidermis becomes the meristematic and forms the cork cambium. And so each cambium produces secondary growth each year. And this is similar to the growth that occurs in roots. Okay, so here we have the primary xylem. Here's the secondary xylem. So this is the one that's adding the actual width to the, the tree or plant. Here's the vascular cambium. It's in between the xylem and the phloem. So the vascular cambium produces the secondary xylem. Okay? And it also produces the secondary phloem. So it increases both ways. When it increases to the inside, which to the inside, and also it adds new cells to the exterior. And then in between or above the cortex, you have the cork cambium, and that produces the cork. So the vascular cambium in roots and stems produces the secondary xylem and the secondary phloem. The secondary xylem is the one that makes up wood, and the secondary phloem makes up the tree bark, the inner part of the tree bark. So here's the secondary xylem that's produced by the vascular cambium. Okay, so this is the wood, and then there's the bark that's part of the secondary phloem, also produced by the vascular cambium. The vascular cambium also produces sclerenchyma fibers and parenchyma cells that are formed in lateral rows of cells called rays. And the production of these tissues is highly asymmetrical, with creation of more secondary xylem that's retained and accumulates as wood. The cork cambium produces the cork cells to the exterior of the trunk, and the secondary phloem, cork cambium, and the cork cells make up the bark of the tree. In order for gas exchange to occur, it has to happen through these small openings that are called lenticels. Okay, so here's a picture of the lenticels. These are small little openings that allows for gas exchange. Here you can get a better picture of how it's increasing in length. So here we have the part where it's just showing you the primary tissues. So we have the pith, the xylem, and the phloem. Okay, the primary xylem and the primary phloem. Then when you have the formation of the vascular cambium here in between those two, this is the one that's going to produce the secondary xylem and the secondary phloem. So looking at this diagram, you can see it in the lighter colors. So the lighter blue and the lighter red, that's indicating the secondary xylem and the secondary phloem. So as it adds more secondary xylem and more secondary phloem, it's increasing in width. Okay. Okay, so we'll pause here and then we'll continue on the next one.